All right. So the fourth and final objective for this packet, the synapse, is to describe the process of neural integration and the role of the axon hillock in this process. So we have previously talked about the different parts of the neuron, and we talked about the soma or the cell body. We talked about the axon, which is the projection towards the end of the neuron, where we have the axon terminal. And more specifically, we talked about the axon hillock, which is really at the neck or the narrowing to become the axon of the neuron. So that region is really important because of a process called neural integration. Remember, we said that the axon hillock is the area where we have an abundance of voltage-gated sodium channels. And so this is the area where the action potential can begin, depending on how many and what type of graded potentials arrive on the soma. Those will all be sort of added up or assessed at the axon hillock. And then that is the area where we decide whether or not threshold membrane potential change to get to threshold has been accomplished, and then we can go ahead and proceed with an action potential. So as far as neural integration, we're looking at a couple of different types of assessments that can be made at the axon hillock. One is summation. And summation simply means we have several different synapses at the soma of the neuron. And so we want to add those together. If we have graded potentials that are arriving that are close enough in time or close enough in space, right? If they're at the same synapse, we can add those together. And so summation is one of the processes that happens, and we determine if threshold has been reached, and then an action potential can happen. The other is frequency coding. We'll talk about what that is. Divergence and convergence, which is a similar idea that we can have several synapses coming onto one neuron, or one neuron synapsing onto several other somas. So divergence versus convergence. And then lastly, the axon hillock and threshold, which is kind of where all the assessment happens. So an action potential is triggered if the membrane potential threshold at the axon hillock is depolarized enough to get us to threshold. So if the potential does not get us to threshold, there will be no action potential. And as we said earlier, even if we have a graded potential that's greater than threshold, it will still be that same action potential in terms of magnitude and amplitude. So the axon hillock is the area on the neuron where we're kind of measuring what does the membrane potential look like here? Because if it is at threshold, then this is the area where we will begin to initiate the action potential. And then through saltatory conduction, if it's a myelinated neuron, we will have that action potential traveling down the entire axon. All right. Now, as far as divergence and convergence, this is just illustrating what that means. So we have a single neuron. Here's the axon of that neuron. Here are the axon terminals. So the axon sort of splits off, which is what that divergence means, into several different axon terminals or axon endings. And so a single action potential could potentially result in graded potentials on each of these individual postsynaptic neurons. So that's what divergence means. We can have a splitting off of the signal to result in graded potentials on different neurons, even though it's coming from a single neuron. The opposite of that would be convergence, and that is just where we have a single soma receiving input from several different axon terminals, right? So again, we can have a multitude of signals coming in that are coming from many different neurons. And then this is what gives us that large network of communication in the nervous system, right? We can have a single neuron getting many different input, and a single neuron also giving off several different signals. Summation, again, basically adding or summing the graded potentials that arrive on the soma or the cell body. And so we can think about temporal summation if it's at one synapse, but it's close enough in time, or spatial summation, which is if we have several different synapses that arrive at the exact same time. So again, graded potentials have the ability to sum. Remember, action potentials don't. So graded potentials can be added to see if it's strong enough to get us to threshold, and then that will ultimately result in the action potential. Now, along with thinking about summation, we want to think about what type of graded potential it is. So remember, graded potentials can be either IPSPs, which are inhibitory changes, or negative membrane potential changes, or they can be EPSPs, which are depolarization, right? Or positive membrane potential changes. So again, when we sum two different graded potentials, it really depends on where they are on the soma, how close in time they arrive on the soma, but as well, what type of graded potential they are. Because if we have two graded potentials, one IPSP and one EPSP, they basically cancel each other out. So even if we're adding them together, because one is negative, one is positive, that will result in no membrane potential change. All right. Now, if we look at the graphics here on the right, in the first panel, we're looking at two EPSPs arriving on the soma, and then an IPSP also arriving. And so if we think about the temporal summation, it's arriving at on different synapses, but close enough in time. So we have synapse A, right? Sorry, this is temporal summation, so it's the same synapse. Excuse me. So it's looking at synapse A exclusively, right? So we have one graded potential arriving at synapse A, and that is an EPSP. Another graded potential arriving, which is another EPSP. And so because they have arrived close enough together in time, then we can add them together, and they're both EPSPs, so that results in a threshold criteria, and so then the action potential is fired. If we look at the panel at the bottom here, this is going to be spatial summation. So now we're going to take into account all of the various synapses that are on this soma, if they come in at the exact same time. So synapse A, synapse B, synapse C, and we're going to add those three graded potentials together. So that's two EPSPs and then one IPSP. And so basically, we're combining those, and that is enough to get us to threshold, so that will fire an action potential. All right, and I think we've talked about summation before. So again, this is a process that happens at the axon hillock that will ultimately determine whether or not the action potential is initiated. The other aspect of neural integration is frequency coding, and it's basically the degree of depolarization at the axon hillock. And so that is going to be regulated by the frequency of action potentials. And we talked about this idea that in the nervous system, and as far as neurons and action potentials go, there is no bigger action potential. There is no action potential that lasts longer or that has a greater amplitude or magnitude. Every single action potential is identical. And so the way that we can grade or send different types of signals is to simply increase or decrease the frequency of the action potential. In other words, how fast are we sending those action potentials? And that will ultimately result in more or less neurotransmitter being released at the synaptic junction. And then that will translate into a greater IPSP or a greater EPSP on the postsynaptic neuron. So frequency coding is the way that we can send different strengths of signals as far as action potentials go. So we can have several action potentials resulting in more neurotransmitter, resulting in a bigger EPSP, or we can have a very slow frequency of action potentials resulting in less neurotransmitter, ultimately resulting in a smaller EPSP or smaller IPSP. All right. Okay. And then we have presynaptic modulation. So this is regulating via synapse how much neurotransmitter is released. And so we have facilitation or inhibition depending on what type of influence the modulating neuron has at the synapse. So here we have a presynaptic neuron we've got our postsynaptic neuron, and then we have this third modulating neuron. Now, this modulating neuron is one, it's forming what we referred to earlier as an axoaxonic synapse. So where we were speaking about the most common type of synapse being an axodendritic, which is the axon terminal synapsing onto the dendrites. Here, this is an example of the axoaxonic because the axon terminal is actually synapsing onto the axon of this presynaptic neuron. The other thing is that the modulating neuron is not directly involved in synaptic communication. It's really important to understand that. So it's not necessarily influencing the electrical activity. It's not influencing the graded potential directly, but it is encouraging the presynaptic neuron to either release more neurotransmitter or to release less neurotransmitter without actually being involved directly in the synaptic communication. All right, so let's look at what an example of that looks like. So on the left is presynaptic facilitation. So here we have our neuron in question. Here we have two different synapses, so synapse D and synapse C, and then neuron E in this case would be our modulating neuron. So neuron E is going to encourage presynaptic neuron C to release more of its neurotransmitter. Okay, and so what that results in is whenever neuron E is active at the synapse, that's going to cause a bigger graded potential at synapse C. So here we have synapse C on its own, synapse D on its own. Notice there's no direct membrane potential change when it comes to E, because again, E is not directly involved in the synaptic communication, and E is not directly going to influence any type of membrane potential change. Instead, all E is doing is it is encouraging synapse C to then release more neurotransmitter, which will indirectly result in a bigger membrane potential change. So if we think about synapse C and E together, so E being that modulating neuron, notice that now it generates a bigger graded potential that results ultimately in an action potential. But if we think about synapse D and E, it doesn't do that, right? Because it's only modulating neuron C, all right? The opposite phenomenon would be presynaptic inhibition. So again, we think about our synapses here, synapse G, synapse F, synapse H, or not synapse H, but H being the modulating neuron, which is ultimately synapsing onto F, but not necessarily onto the neuron itself. So that if we think about synapse F independently, that gives us a small graded potential. Synapse G independently gives us a small graded potential. Synapse H alone gives us zero membrane potential change. So it doesn't at all influence the membrane potential directly or on its own. Synapse F plus H, that's going to result in zero membrane potential change. And the reason is because the modulating neuron H is encouraging or decreasing the amount of neurotransmitter that is released from neuron F, all right? If we were to think about synapse F and G together, they would be able to sum and give us a bigger graded potential that results in an action potential. And then if we thought about all three neurons, right, F, G, and H, basically we will result in a small graded potential as F and H would pretty much cancel each other out. Okay, so this is an example of presynaptic modulation, either facilitation, which encourages more neurotransmitter release, therefore a bigger graded potential on the postsynaptic neuron, or presynaptic inhibition, which is the opposite, less neurotransmitter release, therefore less of a membrane potential change on the postsynaptic neuron. All right, and again, just looking at presynaptic modulation, this is an axoaxonic type of synapse. So it's basically where one neuron encourages or disencourages, right, excites or inhibits another synapse. This is selective. So a modulating neuron will only be modulating one specific neuron. So it's not going to affect the other synapses outside of that single neuron that it is modulating. And then if we think about the axodendritic and axosomatic neurons, these will either excite or inhibit the postsynaptic neuron, and then these will be non-selective. So the way that presynaptic modulation differs from your typical synapse is that the axodendritic and axosomatic synapses are non-selective. As long as it's synapsing onto the soma or the dendrites of the neuron, it's always going to be taken into account as far as neural integration. But for the axoaxonic synapse, it's only going to influence that specific synapse at which it is modulating. All right. Okay. And then just sort of putting all of those pieces together and then bringing in things that we've talked about before. So on the soma of this neuron, we have the presynaptic neuron that is synapsing on to the soma. And so we have several different neurons synapsing onto the soma, and they can be stimulating either IPSPs or EPSPs, depending on what's happening with this presynaptic neuron. Those will all be sort of integrated together, all of the different types of signals, all of the different types of ligands that are binding because of the neurotransmitters released from the presynaptic neurons. All of that is being sort of added and taken into account and assessed at the axon hillock. So this is what neural integration means. We're taking all of that information into account. We're summing what can be summed. We're thinking about the presynaptic facilitation or inhibition that is happening on these presynaptic neurons. And we're kind of adding all of that together, integrating all of it, and then deciding if it's enough to get us to threshold and to result in an action potential. The action potential will then move down the axon hillock, and it will ultimately result in neurotransmitter released from the axon terminal, which will again result in neural integration on the postsynaptic neurons on which these axon terminals are synapsing. All right, so it's really an endless cycle of events that keeps going, again, one direction only in terms of the chemical synapse, and allowing all of our neurons to speak to each other, but also all of our neurons to speak to other types of tissues, right? So our muscle cells, our glandular cells, our cardiac cells, all of those different types of cells will also receive this type of neurotransmitter release and can receive synaptic input from neurons as well.